Hello students, welcome to EPG Pathshala. I am Dr. Soma M. Gurai from Hindu College, University of Delhi. Today we are going to discuss a module, vaccines, which come under the paper Immunology. After studying this particular module, you should be able to understand the concept of vaccines and the various characteristics of an ideal vaccine. You will be able to describe the features of a live vaccine, the live attenuated vaccines, the inactivated vaccines, the toxoid vaccines and polysaccharide vaccines. You have knowledge about advantages and disadvantages of the attenuated and the inactivated vaccines. You understand the recombinant antigen vaccines, which comprises mainly of the viral, bacterial and the DNA vaccines. You know about DNA vaccines in detail, and you learn the newer strategies for the DNA, the vaccine designs. The immune system is a complex system of interacting cells whose primary function is to identify the foreign from the self and eliminate it, usually referred to as antigens. The defense of the body against this particular antigen usually involves the elicitation of both innate and adaptive branch of immunity. The acquired immunity or the specific immunity usually involves the production of protein molecules by the B lymphocytes, also called as antibodies or immunoglobulins, and of specific cells including the T lymphocytes, also known as cell-mediated immunity, whose purpose is to facilitate the elimination of the foreign substances. The passive immunity is the transfer of these antibodies produced by one other humans or any other organisms or animals, and this provides protections against some infections, but this protection is temporary. Active immunity is the stimulation of the immune system to produce antigen-specific humoral and cellular immunity. That is basically the elicitation of the B and the T lymphocytes. Now, unlike passive immunity, which was temporary, active immunity usually lasts for many years, often for a lifetime. Vaccines interact with the immune system and often produce an immune responses similar to that produced by the natural infection, but they do not subject the recipient to the disease and its potential complications. A vaccine is defined as a preparation of bacterial, viral, or any other pathogenic agents or their isolated antigens. Now these can be peptides, DNA, RNA or even polysaccharides, which is administered into the objective to elicit the immune responses within the recipient body. Primary immune responses towards vaccines leads to formation of the IgM type of immunoglobulins, leading to activation of both B and T lymphocytes, as well as to the formation of the memory cells. But the secondary response towards vaccines is due to the memory cells, which is rapid, and the response to the pathogen is by the high affinity IgG or immunoglobulin G type of immunoglobulins. Vaccine therefore is basically an antigen or its components that can induce the secondary immune responses in the host. We can broadly say that a vaccine aims to introduce immunological memory against a pathogen in the host. Primary and secondary immune responses to vaccines. It can be seen here that the primary responses to vaccines has an increase in the IgM production, and the IgG is also seen, which is the memory cells which have been developed here. And in the secondary responses to vaccines, high amount of IgGs can be seen, which are high affinity IgGs, to which the later responses can be observed. A chapter on vaccines cannot be completed without mentioning the pioneering work of Edward Jenner and Louis Pasteur. In 1798, Edward Jenner privately published a small booklet entitled An Enquiry into the Causes and Effects of the Variola Vaccinae, a disease discovered in some of the western countries of England, particularly Gloucestershire, and known by the name of cowpox. The first pioneering work for vaccines is hence attributed to Jenner, who used inoculation on an 8-year-old boy named James Phipps, who was suffering from smallpox using cowpox pustules. Talking about Louis Pasteur, he is revered by his successes in both the life sciences as well as by the general public. In fact, his name provided the basis for a household word pasteurized. He demonstrated that microorganisms cause both fermentation and disease. He developed the earliest vaccines against fowl cholera, anthrax, a major livestock disease that in recent times has been used against humans in germ warfare, and the dreaded rabies. He was the first to officially vaccinate a 9-year-old boy, Joseph Meister, who was repeatedly bitten by a rabid dog. Meister later became the gatekeeper of Pasteur crypt. The characteristics of a good vaccine involves 1. A vaccine should be able to generate immunological memory. Both T and B lymphocytes should be formed, hence should be able to generate both arms of immunity, that is, humoral as well as cell-mediated immunity. Second, it should have the ability to generate the appropriate immunity like cell-mediated immunity should be developed for tuberculosis and viral pathogens, while humoral immunity for all the other bacterial pathogens. Third, it should be able to provide lifelong immunity with a single dose. Fourth, it should be able to be introduced to the recipient, probably through a non-invasive method like through oral administration or nasal spray. 5. Vaccines should be inexpensive, easily manufactured and stable in extreme temperature or humidity. 6. It should be easy to store and transport. Types of vaccines. There are many types of vaccines, and the vaccines that are used are: number 1, natural life vaccines. Cowpox vaccines are used against smallpox. Simian rotavirus used against gastroenteritis. Second, live attenuated vaccines. BCG, Sabin polio vaccines, MMR, varicella zoster vaccines, yellow fever vaccines, etc. For inactivated vaccines, Salk vaccine, polio vaccines, rabies vaccines and pertussis vaccines are involved. For toxoid vaccines, diphtheria vaccine, tetanus vaccines are seen. For polysaccharide vaccines, HIV vaccine, pneumococcal, pneumonia vaccines, meningococcal vaccines are been solved or seen. For recombinant antigen vaccines, HBs antigens vaccines have been produced. For viral vector vaccines, HIV vaccines with ENV, GAG and POL genes inserted in many canary pox viruses. Second, hepatitis B vaccine. Third, influenza antigen inserted in vaccinia virus vectors. For bacterial vector vaccines, attenuated Salmonella typhimurium as vectors for cholera vaccine has been used. For DNA vaccines, rabies vaccine, influenza vaccine and HIV vaccine has been seen. The natural life vaccines. Now these vaccines include preparations from live non-pathogenic organisms, but that still have the ability to elicit immune responses. The classic case has been the use of cowpox viruses. Whereas the live attenuated viral vaccines, attenuated means to weaken, is a process through which the pathogen loses its virulence but becomes incapable of causing a disease. The process involves growing the microbes. It can be bacteria, it can be viruses, in high bile concentrations, reduced temperatures, repeated subcultures, or even passes through foreign cells or tissues like embryonated cells and eggs, etc. Attenuated bacteria such as Mycobacterium tuberculosis is used as a vaccine called BCG or Bacillus calmette-guerin against tuberculosis, and the viruses like polio vaccines, yellow fever and measles have been the most successful. The advantages of the live attenuated vaccines are: one single dose is capable of inducing long-term immunity, it provides wide spectrum of immunity, it has a rare occurrence of any allergic reactions or post-vaccination slumps, and it is cost-effective. But the disadvantages are also there. It has the potential to revert back to the virulence form. It can exacerbate the diseased conditions in immune-compromised individuals. and in some rare cases, it may lead to abortion and infertility. Inactivated vaccines are those vaccines which are prepared by inactivating or killing the pathogens by heating or chemical treatment, like the use of beta-propionolactone or formaldehydes. The process usually changes the surface antigenic determinants which may not be effective against the natural pathogens. The advantage being heat stable, can be administered into the immunocompetent individuals, and there is no danger of mutations or reversal to the pathogenic forms. Now, these kind of vaccines stimulate humoral immunity but are very low on cell-mediated immunity, and thereby they require repeated booster doses and have high costs. Inactivated Salk polio vaccines and rabies vaccines are good examples of inactivated vaccines. This slide shows how toxoid vaccines have been developed. Some bacteria are pathogenic due to the extreme exotoxins as potent cytotoxic microbial poisons, causing severe infection. Toxoids are modified toxins. These are exotoxins that are incapable of virulence but can elicit immune responses. Toxoids are generally generated by treating the exotoxins with higher heat or chemicals like formaldehyde. Vaccinations by toxoids produces antibodies which can bind and neutralize the pathogens. Vaccines against diphtheria and tetanus are produced from toxoids. Disadvantages of toxoid vaccine is that not all bacteria produce toxins, and even if they do, it is in minute quantity which is difficult to isolate and characterize. This slide shows the polysaccharide vaccines. Bacterial polysaccharides are the classic antigens for B cells and are recognized by the BCR that has the correct specificity. Interaction between the polysaccharide and the BCR induces signals that are sufficient for clonal selection and antibody production, but without immunological memory. Polysaccharide protein conjugate vaccines interacts with BCR and elicit T helper cells by antigen presentation of the protein component to the CD4 positive T cells, thereby inducing memory B and T cells. Therefore, this strategy has been exploited to produce pathogen-specific vaccines that target bacterial polysaccharides. The first conjugated polysaccharide vaccines was for HIV. The recombinant vaccines. The immunogenic proteins of pathogens can be synthesized artificially by introducing a gene coding for that particular protein into an expression vector that can be either virus, yeast or bacteria, anything. Now, such products are sometimes referred to as recombinant vaccines. For example, in hepatitis B, human papilloma viruses and influenza vaccines, these are basically produced by the recombinant vaccines techniques. This slide shows the viral vector vaccines. Now, this slide shows how a viral vaccine can be generated by using a harmless virus, but the protein, when inserted into a vector, can be used for the vaccine production. Here it has been seen that the harmless influenza virus causing no disease is recombined with a viral vector such as adenovirus and alpha virus, etc. The protein for hematoagglutinin HA is inserted into the virus vector, which then expresses both the proteins for the adenoviruses as well as hematoagglutinin HA of the influenza virus. Now, this protein is directly injected into the muscles of the humans, which results in stimulation of both humoral and cell-mediated immunity. A classic example of using viral vector vaccines in bacteria as a vector is seen here, where hepatitis B recombinant vaccine is produced. The viral protein is expressed in bacteria, yeast and other mammalian cells and even viruses, which we have seen in the previous slide. Here we see that the DNA cloned and the plasmid DNA is cut with the same restriction endonucleases. Then these fragments are mixed, spliced and ligated. The plasmids contain the different inserts are then mixed to the bacterial culture and transformed. Each colony of the bacteria is then grown for the specific gene library, and the culture of the host bacteria now have the desired bacterial protein to be inserted into the humans or directly into any of the host for vaccine production. Now, as we have seen the viral vector vaccines being produced and used as vectors for viruses as well as bacteria, here we see another classic example where the virus vaccines are generated into the viruses, a different virus, and express a foreign gene. You can see here very classically that the plasmid, which shows the vaccinia promoter as well as the thymidine kinase gene, is cleaved by the restriction enzyme, and the DNA encoding the antigen from the pathogen is inserted into this particular site. Later on, this plasmid is ligated with the desired DNA encoding the antigen. The gene of the pathogen with the recombinant plasmid is then transfected into the tissue culture along with the vaccinia virus. That is the infection part. The homologous recombination occurs, and then the culture is selected using bromo-uridine selection, and finally the recombinant vaccinia vector vaccines is obtained, which the recombinant vaccines can then multiply in the infected cells and produce the antigen a wide range of different viruses. We see not the virus as the vector but bacteria as the vector for the production of vaccines. Similar to the viral vectors, some bacterial strains are engineered to carry genes of virulent pathogens. The DNA encoding the antigenic determinants is inserted into the attenuated bacterial genome. The bacteria then expresses the antigen along with its proteins. This expression of foreign antigen stimulates the immune response. Here, a classic example is given for the Salmonella typhimurium T21A, where the antigenic proteins from the wide array of virulent pathogens is cloned into the live attenuated bacterial vectors. The cloning of the gene encoding the protective antigen is then transformed into the live attenuated bacterial vector. The plasmid that encodes that vector gets inserted into the chromosomal DNA of the bacteria. It is intracellularly expressed as well as surface display of certain signal peptide mediated translocation occurs, which can be seen on the membrane or the flagella bounded antigen. They are also expressed as secretory antigens via various secretory systems. Now, these expression of antigenic proteins by either the constitutive promoter or induced by the environmental stimuli using an in vivo inducible promoter, for example, in the case of oxygen depletion, results in the elicitation of the humoral immune responses, that is via the production of the antibody, or elicitation of the cell-mediated immune responses, that is via the elicitation of CD4 positive or CD8 positive T lymphocytes. Now, this gives rise to protective immunity against the target pathogen. Apart from viruses and bacteria, DNA can be itself used to elicit the immune responses. DNA immunization is a novel technique by which direct injection of genetic material into the living host causes a small amount of its cell to produce the introduced gene products. After immunization, transfected muscle cells may produce antigen or foreign proteins that then directly stimulate B cells of the immune system, which in turn produce antibodies. Direct transfection of antigen-presenting cells, that is, either dendritic cells or macrophages, with subsequent processing and display of MHC antigen complexes may also occur. Production of DNA vaccine starts with E. coli transformed with the plasmid of interest. The transfection is done into the myocytes, which results in the both humoral and C-mediated immune responses. So here it can be seen that the muscles secretes the secreted antigens, which in turn produces humoral responses. It can also elicit or excite the antigen-presenting cells, which in turn promotes or proliferates the cytotoxic T lymphocytes, that is THCR MHC1 recognition, or also can recognize and elicitates the T helper cells via the TCR MHC2 recognition. Shows the advantages of using DNA as vaccines. DNA vaccination has many advantages compared with the conventional vaccine approaches, particularly in the setting of protecting against potentially lethal emerging infectious diseases. DNA vaccines can accommodate a combination of different genes that encode or code for different antigens from one or more different pathogens. This can result in the generation of broad immunity to multiple protein antigens. DNA vaccines have also been observed to stimulate both antibody and T cell arm of immune responses, including those that are specialized to kill viruses and cancers via the cytotoxic or killer T cells. A significant advantage, especially for emerging pathogens, is that DNA vaccines do not require the handling of potentially deadly infectious agents. Lastly, in addition, they can be easily significantly produced within a very short production time. Now this slide shows a table on the different vaccine strategies and the diseases against which it has been explored. Number 1. Synthetic vaccines. Diseases which are explored are peptide sequences used to prepare vaccine for AIDS, malaria, schistosomiasis, hepatitis B, and the use of multiple peptides increases immune responses can be observed. It has poor immunogenic owing to improper folding, which is the main characteristics of the naive antigen. The first synthetic vaccine was developed in 1999 against hemophilic influenza type B. Second type of vaccine strategy can be the microencapsulation delivery system. It has been seen for antigen kept inside the capsules in the form of microspheres or nanospheres and delivered inside the body. This capsule is made usually of lactic or glycolic acid. Now, vaccines for influenza has been produced by this method, but it lacks cell-mediated immunity. Third strategy has been the solid matrix antibody antigen complexes formation. Monoclonal antibodies were raised against the antigenic determinants and later saturated on a solid matrix. The complex, that is the matrix antibody antigen, is then delivered in the body. Clinical trials for many infection diseases are carried out for this particular strategy, and it has been seen that it stimulates the T helper cells. Fourth strategy, the liposomes and the micellates. Liposomes are phospholipid bilayer with an aqueous interior. They are used to enclose antigens and delivered. Antibodies are raised once the liposomes are fused with the cells and antigens released in the cytosol of cells. For example, hepatitis A vaccines have been produced via this method. It was first discovered by Professor Junzo Tsunamoto in 1992. Next strategy is known as ISCOMS, which is the immune stimulating complexes. This is basically a mixture of lipid and detergents and a glycoside derivative which is used to encase antigen and introduced into the host. Vaccines for HIV and measles are being considered in this particular strategy. Next strategy is the antigen anti-idiotypic vaccine. Anti-idiotypic antibodies mimic the antigen binding sites, that is the idiotypes of the antigen, and when such antibodies are introduced in the body, they recognize the antigen and clears it. Hepatitis B vaccines are being produced via this method. Last strategy is being used as known as antigen cochleate process. Now, phospholipid bilayer and calcium complexes is prepared on which antigens is rolled like sheet. Once delivered, the antigenic peptides escape the degradation and are able to elicit both humoral as well as cell-mediated immunity. Clinical trials for many of the infectious diseases are being considered via this method. So students, let us summarize what we have learned in this module. Firstly, a vaccine is a preparation of pathogenic agents or their constituents that is administered into the host to elicit immune responses. An ideal vaccine should provide lifelong immunity, be non-invasive, stimulate both humoral, that is the B cell mediated lymphocytes, to produce the antibodies, as well as cell mediated immunity, or that means it should elicit the T cell lymphocytes, that can be both cytotoxics and the T helper cells, to develop the immunity. Should be cheap, should be easy to store and transport. Vaccines commonly can be used are the attenuated, the inactivated, the toxoid and the polysaccharide vaccines, which are very common these days. Now, what do you mean by attenuated vaccines? Now, these are weakened pathogens administered to stimulate the immune responses in the host body. Inactivated vaccines were the one that are killed pathogens, while toxoids are the one that are chemically modified pathogens that have lost their toxicity but still regain the immunogenicity. Now, capsular polysaccharides are also being used to generate immunity, and vaccines have been made to show with good results. The most promising form of vaccine design are the recombinant antigen vaccines, which introduces a gene coding for the antigen into the host cells. The bacteria and the yeast can be used here using the recombinant DNA technology. Now, this protein coded is expressed, harvested and can be used as vaccines. The live vector vaccines, whereas the bacteria or the viral serves as vectors and the source of antigens inside the vaccine. The antigen genes are introduced in the attenuated bacteria or harmless viruses and are infected back to the host system. The antigens formed and released by these vectors thereby stimulates both the T cell and B cells, that means stimulating both the humoral and the cell-mediated immunity in the body. The DNA vaccines show much promise these days because they involve direct introduction of the antigen gene into the muscle cell or the antigen presenting cells. Now, once inside, the gene is expressed and the antigen displayed along with the major histocompatibility complex on the host cells, stimulating both the humoral and cell-mediated immunity. Thank you.